Hello everyone, I will introduce our study on Lake Bosten, asking how a warm dry to warm wet shift reshapes lake environment, microbiomes, networks and functions, and whether functions remain buffered. Northwestern China has become warmer and wetter over 6 decades, altering lake water level, connectivity, salinity and nutrients. Microbial responses across diversity, stability and function remain unclear. The graphical abstract shows that warm wet, higher water level, and lower salinity and total nitrogen triggered community reorganization, assembly shifts, network rewiring and nitrogen redistribution. Key message: strong taxonomic and interaction turnover, but buffered broad functional potential. We combined long-term records from 1958 to 2022, decade-separated field surveys with 16S 18S amplicons and metagenomics, and a 40-day mesocosm experiment with temperature, salinity and nutrient scenarios. Long-term records showed basin warming, renewed precipitation after 1987, and a negative water level TDS relationship, indicating warm-wet dilution. Warm-wet conditions had higher water level, lower TDs and TN, and higher water temperature and chlorophyll A, suggesting habitat reconnection and reduced dispersal barriers. Bacterial diversity increased significantly, whereas eukaryotic diversity increased weakly. Bacteria were linked mainly to TN, water level and TDS, eukaryotes to TN, water temperature and water level. POCA showed clear bacterial and eukaryotic separation between periods, indicating community restructuring. Variocomicrobiota and cryptomycota increased, while firmicutes and diatomea decreased, showing replacement among ecological groups. Homogeneous selection increased and dispersal limitation weakened, consistent with higher connectivity and stronger shared environmental filtering. Within domain, bacterial and eukaryotic networks became less complex and stable, suggesting more fragile internal interactions. By contrast, the bacteria-eukaryote network strengthened, with nodes increasing from 34 to 153 and edges from 91 to 1937. Thus, within domain stability weakened, while cross-domain coupling strengthened and may support functional buffering. Broad KEG and COG functional composition showed no significant period-level shift, indicating buffered overall functional potential despite structural reorganization. At finer levels, more genes were enriched in warm-wet samples. Assimilatory nitrate reduction increased from 39.7% to 65.5%, while denitrification decreased from 47.9% to 13.3%. Thus, function changed through selective pathway redistribution, not simple gain or loss. SEM linked warm-wet environmental conditions to diversity, stability and redundancy through water level, TDS, water temperature, TN and chlorophyll A. Diversity was negatively related to compositional stability but positively related to functional redundancy, suggesting greater buffering despite less stable composition. This supports buffering by functional redundancy, cross-domain coupling and guild replacement during microbial restructuring. Overall, the study connects climate, environment, communities, networks and functions. Warm-wet transition changed connectivity, dilution, salinity and nutrients, reorganized communities, weakened within domain networks, strengthened cross-domain coupling, and redistributed nitrogen cycling. In conclusion, arid lake microbiomes were strongly restructured, but broad functional potential was maintained through redundancy, coupling and guild replacement.